Okay, good day, everyone. Just as I promised, in this study session 3, we are going to talk about enzymology. And here we'll talk about enzymes, the cofactors, naming and classification of enzymes, and then we talked about the general properties of enzymes. Okay, what are enzymes? Enzymes are chemical substances, usually biomolecules that are used in catalyzing the biochemical reactions or chemical reactions rather. So you have a chemical reaction. We know in the chemical reaction, we have what happens in chemical reaction is basically bond breakage and bond formation. You have a bond being broken. The energy released in breaking a bond is used to produce a new bond, usually. So that is why when that energy that is released due to breakage in the bond is enough to form a bond, there is no need for external energy to be added. For example, even when the energy is even too much, you can have, for example, a particular bond being broken and the release energy, for example, say 25 kilojoule, and the new bond to be formed only need 10 kilojoule or 15 kilojoule. You can see that is an excess of 10 kilojoule. That type of reaction, we call it. Can somebody tell me, Miriam? Miriam, are you with me?
Sir.
Can you hear me?
Yes, sir, I can hear you.
Remember, what do you call this? Exothermic and endothermic reaction?
Sir, you see?
Can you remember exothermic and endothermic reaction?
Yes, sir.
Yes. So in a reaction, remember I just said that in a reaction, what usually happens is you have bond breakage and bond formation. When you have a chemical reaction, there is a breakage in a bond of the reactant. For the product to be formed, then there is the bond formation. So if that energy, a bond breakage causes only the release of energy. If that energy is enough to form the new bond, is even more than enough to form the new bond, then you can have some of this energy being used to form the new bond, while the excess energy is then released into the surrounding. And this is what we call exothermic, right?
Yes.
While in the case of endothermic, it is a reaction in which the energy is needed from the surrounding. So for example, when you break a bond that has 25 or that release energy of 25 kilojoule, and you want to form a new bond that needs 30 kilojoule, you can see that the energy that was released is not enough to form the new bond, right? So you need an extra energy for that new bond to be formed. So that extra energy must come from the surrounding. So what happened in the case of enzyme? An enzyme usually play important role in lowering the energy needed for reaction to take place. So when you have a reaction that need a 25 kilojoule, an enzyme facilitates the process in such a way that you will end up using maybe 15 kilojoule. Is that clear? So the role of enzyme is to lower the energy required for bond formation. When we go back a bit to history, we can see that there are a lot of several processes. We don't need to mention here because of time. Several processes and activities of which the activity of enzymes were witnessed. But the first enzyme was isolated in 1926 by James Sumner. And what enzyme is that? That enzyme is urease. Yeah, I think when we are doing the metabolism, we will know what role does the enzyme urease plays. So another thing we need to know about enzymes is that basically all the enzymes are proteins, but there are what we call, with the exceptions of what we call ribozymes. The ribozymes are not proteins. They are RNA. They are RNA enzymes. They are RNA enzymes. They are not proteins. So with the exception of ribozymes, we can say all enzymes are proteins. Another thing we also need to talk about is that an enzyme contains what we call an active site. Contain what you call an active site. Within the active site of an enzyme, there are some amino acids. We call them the active site residues. These active site residues play a very important role in substrate conversion. They are playing important role in converting the reactants to products. So in the active side, we can see the amino acid residues. And in addition to amino acid residues, there are also some non-protein substances that also help in converting the substrate to product. They are called cofactors. A cofactor is called a coenzyme when it is an organic compound, usually a derivative of vitamin, loosely attached to the enzyme. So when you have a coenzyme that is loosely, you have a cofactor that is loosely attached to an enzyme, and it is an organic molecule, which we said usually a derivative of vitamin, it is called a coenzyme. When you have a coenzyme that is not loosely attached, it is tightly attached to an enzyme, either through covalent or non-covalent interaction, it is called a prosthetic group. So it is a coenzyme. Usually coenzymes are loosely attached. But when you have a situation that they are tightly attached, maybe through covalent or either non-covalent, they are called prosthetic group. When that cofactor is not an organic, rather it is inorganic, usually a divalent metal or even trivalent metal like iron 3, it is called a metalloenzyme. A metalloenzyme can be, when you have a metalloenzyme without the metal, it is called an aphoenzyme. But with the enzyme, it is called a holoenzyme. So properties of an enzyme. The enzyme has a high catalytic power. Just like we said, you can have a biochemical process. There is the possibility of when you have reactant and the two reactants together, they can react without an enzyme. On their own, they can react. But the problem is you can have maybe they can take 20 minutes for them to react, for example. But when you have an enzyme, they can take only 1 second to react. So they have the ability to catalyze, they have the power to make the reaction go at a faster rate. Another thing is enzymes are highly specific. They are highly specific in the terms of the reaction they catalyze and in terms of the substrate. And this specificity can be in four form rather. Can be absolute specificity, can be group specificity, can be linkage specificity, can be stereochemical specificity. In absolute specificity, you have an enzyme being specific to a particular reaction and to a particular substrate. You will see a particular reaction that this reaction can only be catalyzed by this particular enzyme, or this particular substrate can only be converted to product by this particular enzyme, only, no any other. But you can have some enzyme that can catalyze a group of substance reactants For example, see, maybe you have this reactant and it contains a particular functional group. So once that functional group is there, so that enzyme can easily convert it to another substance or to a product. You can have also an enzyme that are only specific, for example, to a linkage. In our previous class, we talked about trypsin, chymotrypsins in the process of discussing sequencing of a protein, where we said a trypsin is specific to some amino acid residue. So even the amino acid residue, they are specific to either the carboxyl or the amino chain. So we say trypsin. I think if you can remember, we say trypsin cuts across arginine, tryptophan, and another amino acid. And we said even these amino acids, when they locate these amino acids, they don't touch anywhere. They only cut the carboxyl chain or the carboxyl group of that amino acid. So they don't touch the amino group of that amino acid. So the bond formed between the carboxyl group of that amino acid and the other amino acid, that is the bond that this enzyme only cut. It will not cut any other than that. So enzymes are very specific in that. And with regard to stereochemical, we made mention that while talking about the monosaccharides, that most of the naturally occurring monosaccharides, they are in D form. And we said that even the amino acids that occur in the biological systems, they only act on D form. So you can have some amino acids that are stereospecific. They can only, for example, combat D glucose, but not L glucose. So the next thing is that enzymes do not alter equilibrium. Just as we mentioned, they don't change the equilibrium. The reaction can reach equilibrium. It only make the reaction faster. But once the reaction reach equilibrium, the enzyme will not push the reaction to one end. The activity of an enzyme increase with an increase in temperature. We can explain this here, that the increase in temperature increase the activity of an enzyme. And we know simply how increase in temperature cause an increase in chemical reaction due to increase in collision factor. And increase in collision factor, we know it facilitates the coming together of the enzyme and the substrate, and hence the conversion of the product of the reactants to the product. So this is the same way temperature causes an increase in the enzyme activity. But when the temperature is too high, it can actually cause denaturation of the enzyme. Therefore, the enzyme can lose its own tertiary structure. Therefore, so at a particular temperature, you will have the maximum activity of the enzyme. But when the temperature proceeds, then it can cause damage to the protein. As such, it can affect the protein activity. So the same thing also with regard to pH. Extreme pH is bad, either it's alkaline or acidic pH. So enzymes function as specific pH, even though you can have some enzymes like pepsin being can be so active at or being optimum at acidic pH, and you can have an arginase found in the liver being optimum at basic pH. So also with regard to the concentration, as the concentration increase, the activity of the enzyme increase, unless when the enzyme reaches saturation point where you have the increase in the substrate, do not cause any increase in the activity of the enzyme. At that point, you have the rate of the reaction will be very maximum. That is what we call a maximum velocity. And at that maximum velocity, we have the substrate concentration called Km. That is Michaelis constant. For those of us that will do derivation of Michaelis-Menten equation, they will definitely have a much idea on this. So the next thing is the nomenclature of enzymes. Initially, names were given to enzymes based on the individuals that discover these enzymes, or by adding a suffix like ASE, to the name of the substrate, like lipase for lipid, nuclease for nucleic acid, peptidase for peptides, etc. But this naming pattern helped to give us sufficient information with regard to the type of reaction and the substrate involved in a reaction. Therefore, there is a need to come up with a new way of naming an enzyme. So in 1961, the International Union of Biochemistry developed a new way of classifying enzymes. They classify the enzymes into 6 classes, and they assign a 4-digit number. So the classification was done in such a way that the 4-digit number, you have a class, subclass, subclass, and then the individual enzymes. So 4-digit number we are given to represent each. For example, the first number represent the class, the second digit represent the subclass, the third one represent the subclass, and the fourth one represent the individual in them, which we will see on this based on the classification. So we are going to see through the 6 classification. The first one are auxiliary reductase, the second one are transferases, the third one are hydrolysis, lyases, isomerases, and ligases. What are oxidoreductase? Yeah, oxidoreductase. They are enzymes that are involved in oxidation reduction reaction. We know all what is oxidation reduction reaction, involving the transfer of electrons and hydrogens, transfer of oxygen as well. Then we have a donor, we have an acceptor. Example of this enzyme like oxidases, oxygenases, peroxidases, etc. And for example of peroxidase, we have a catalase. Catalase is involved in the conversion of hydrogen peroxide to water. And we can see the enzyme oxidoreductase is 1. The class is given 1. The enzyme commission number, therefore 1 indicates that that enzyme is an oxidoreductase. The other one means the subclass. Another one is the sub of that enzyme. Then 1, it is the name of that enzyme. Here in this case, we have alcohol dehydrogenase. The second one, we have transferases. These enzyme transferases are involved in catalyzing the transfer of chemical group from one molecule to another. We can have an amino transfer, acyl transfer, phosphorus transfer, glycosyl transfer. For example, we have amino transferase or hexokinase. In hexokinase, we have a phosphorus transfer of phosphorus from ATP to a hexose. In hydrolase, hydrolase is an enzyme that involved in the catalysis, that catalyze hydrolysis reaction, the addition of water that end up in breaking a bond. We have a lyases. The lyases, they are involved in catalyzing reaction in which a group is removed to form a double bond or is added to a double bond. We have example of the carboxylase, the hydratase, etc. We have isomerase, a classes of enzymes that are involved in the moving a group or a double bond within the same molecule, just changing the direction or changing the position of the double bond or changing the position of a particular group. We have epimerase, we have resmase, we have mutase. For example, in resmase, we have D and L isomer. We talked about D glucose and L glucose. So these enzymes are involved, isomerases are involved in the conversion between the D and L isomers. We have ligases. These classes of enzymes involved in catalyzing the bond formation between two substances, two molecules. In this reaction, in most cases, an energy in form of ATP is needed. So you can have the bond hydrolysis of ATP leading to formation of a new bond, which we earlier explained in the while trying to explain the activity of an enzyme or enzyme catalyzed reaction. So significance of enzymes, clinical significance. We made mention that in clinical diagnosis, some enzymes have been used. I think you will know this better. You can tell me some of these enzymes that have been, the activity being determined in some certain organs, because we have what we call isozymes. So you can have a particular organ expressing the same enzymes, but differently. And the activity of these enzymes determine the status of that organ, whether that organ is in healthy status or not. For example, in the case of myocardial infarction, where we have an increase in the level of creatine kinase and lactate dehydrogenase, and when a doctor asks for the analysis of this, you know that this is what he is trying to check. So some of these enzymes are used in the clinical settings to determine the healthy state of some organs that play a very important role in disease diagnosis. We can see in heart and kidney, we have lactate dehydrogenase. Also lactate dehydrogenase, we can see a red blood cells in heart, kidney, and brain. We can have a lactate dehydrogenase in brain, lungs, white blood cells, skeletal muscles. They can all be used to detect the level of any damage, any problem in these organs. Mariam, are you following me?
Yes, sir, I'm following you.
Okay, so this is what you need to know about this study session 3. And like I said in study session 4, I already gave an assignment on that, where some of you will talk about some aspect of enzyme kinetics, where I think I have a group that will talk about the activity of an enzyme. Please, you need to know what is an enzyme activity. Well, this is general now, not only specific to the group that need to talk about the enzyme activity. You need to know what is an enzyme activity. What is the unit of an enzyme activity? You need to know how do you differentiate between enzyme activity and specific activity. You need to know turnover number, maximum velocity, and Km. So for those of us that will also, there is a group that will talk about derivation of Michaelis-Menten equation. Please know how to derive Michaelis-Menten equation and what are the various linear form of presentation of Michaelis-Menten equation. We have Line-Weber-Burg plot, we have Adie-Hofstede equation, we have Wolff equation. Please, you need to cover that. The assignment also need to cover that. In study session 5, enzymology 3, the assignment is basically on enzyme inhibition, where you will classify enzyme inhibition as reversible or irreversible. Some will talk about different types of reversible inhibition as competitive inhibition. Another group will talk about uncompetitive inhibition. Another group will talk about non-competitive inhibition. But basically, everyone needs to know about competitive inhibition, uncompetitive inhibition, and non-competitive inhibition. And there are various plots. How do you plot these competitive, non-competitive, and uncompetitive inhibitions? And then another group will talk about regulation of enzyme activity. One will talk about allosteric due to non-catalytic binding. Then another group will talk about covalent modification. And you need to know what is allosteric inhibition, what is covalent modification, how do you differentiate between the two? And okay, this is the aspect that, I thought this, I already discussed about the clinical application of enzymes in diagnosis. So another group will talk about that. And if possible, we can also include the importance of these enzymes in drug development. Mariam, do you have a question?
Yes, sir.
Okay, go ahead.
Sir, in our previous class, you talk on amino acid composition of protein.
Yes.
Sir, please, in the steps, I think it's the third step that you talk on detection. You talk on amino acid is being reacted with. Is it ninhydrin? But
Yes.
I'm not sure. You talk on, is it ninhydrin or choline?
Ninhydrin.
Yes, sir, you give color, but you see it gives a color of blue, and you also talk about another thing. You see it also give color of, is it yellow, orange?
Yellow. Yellow.
So, sir, what's that?
Specifically, all the amino acids react with ninhydrin to give either a purple or blue color. But in the case of one amino acid called proline, when it react with ninhydrin, it gives a yellow color.
Okay.
Yes, that's the only difference.
Okay, thank you, sir. That's I need a fish.
Yeah, right.
Thank you.
Fatima, are you with us?
I'm with you, sir.
Do you have a question?
No, sir.
Okay. The other person with us, are you with us?
I'm with you, sir.
Do you have a question?
No, sir.
Okay. So please, you will. I want to believe you've started the assignment, and I've given you 2 weeks for the assignment. Please try and get the assignment ready, in both the individual assignment and the group assignment. I don't know. Do we pick tests, or do we suggest the time for test, or there is the plan that the university follow?
I'm not sure there's a plan by the university for the test, sir.
Normally,
I'm not sure of that.
Have you written any test for any course?
No, sir.
So far?
Not at all.
Okay, so what we'll do, I know that there is a test component which carry, I know there is a test component which carry 15 marks. So what I will do, is somebody talking? I cannot hear you. Your voice is breaking. Is somebody talking?
Oh, nobody's talking.
Okay, because the voice is broken. So what I'm saying is, I learned that there is a test component that carry 15 marks. So I'm not sure how it is done. So I've already given you the assignment, which carry the 5 marks, and the other one, I think, which carry 15 marks. I was told there is a test component also that will carry 15 marks. So when I confirm that, I will find out and communicate with you the nature of the test, which is CBT, you know. So we'll find a way to, I think we'll discuss through the WhatsApp group so that we'll know what we are going to do and how we are going to, we're going to do that, okay?
Okay, sir. There is still quiz. So.
Okay.
Hello, sir.
Yes?
Quiz.
Okay, that is the test. I think that's the test I'm talking about, right? How is it done?
No, I don't think quiz is. It carries I guess quiz is 5 mark. The individual assignment is 15 mark.
Yes. The quiz you said it carries how many marks?
I guess it's 5 mark. I guess it's 5 mark.
And what is the nature of the quiz?
Just OBJ. Like, what is this? A, B, C, you pick from it. It's an OBJ question.
Five
so
marks.
maybe 5 questions, 10 questions, 20 questions, depending on how the tutor will set the question.
Okay, but it carry 5 marks.
Yes, but that individual assignment is 15 mark.
Yes, I know that. No problem. Then we'll discuss when we are going to have the quiz then.
Okay, sir.
Maybe through WhatsApp group.
No, sir. I think the quiz
They will just paste
is
it.
on the LMS.
It's LMS.
Okay, we can paste it there, open.
Yes.
There is no time.
There's a way they paste it, sir, that it's even some are 2 attempts, while some are just 1 attempt. And there's a time duration for the quiz. So if it's 15 minutes, once it's in 15 minutes, the quiz will allow for that person that already started it.
Okay, I got it. So what I will do is I will find out how it is done so that I will just do that. So I will notify you.
Okay, sir.
Okay,
Yes,
sir.
yes.
Okay, sir.
Do you have any question?
No, sir.
It's all right. So thank you very much. See you another time.
All right, sir. Thank you so much, sir.
Thank you so much, sir. Bye.
All right.